Hi everyone. Thank you for joining today. I'm quite excited to sort of cover another sort of CSCE sort of exploration club session. So thank you for joining whoever has joined in the previous sort of analysis that we've performed on different case studies and including this new case study for this week. So for this sort of single cell exploration club, I'll try to sort of walk you through a different type of analysis, a different disease model, and probably different features that what like you as a user haven't really encountered or have used in the bioterrain ecosystem or in BBrowserX Pro. So sort of the main focus of today will be how we can reanalyze a data set that we can mine from the talk to data system, specifically relating to a unique type of sort of serial single cell data set, so longitudinal single cell relating to stage 4 breast cancer, and particularly specific sampling sites that are metastatic. So the study that we'll use will be the Otsuji data set, and I'll show you how you're able to find that study in the system. So mainly the focus will really be to really uncover how metastatic precursor and drug-tolerant cancer cells evolve. Do they persist during therapy? And do we see some changes relating to the sort of trajectory or developmental trajectory of these specific cells across different clinical indications? So we'll try to sort of answer those questions using the CNV for CNV based cancer cell prediction sort of feature that we have in the BBrowserX Pro system. From there, can we then subset those cells even further using our meta-reference cell type prediction algorithm and sort of querying specific genes where we're able to visualize them within the UMAP and also other visualization tools. And can we then even subset further by running pseudotype mapping to really get an understanding on those trajectories? So that will sort of be the focus of today, relating to these different features, and also how can we define those molecular identities within these metastatic precursor clone cells. Okay. So I'll go into the next. So just before I go into the actual case study, I do want to mention that if you're new to these exploration club sessions, we do hold them every week. We try to cover a vast sort of list of different topics, whether that's relating to oncology, tumor heterogeneity, inflammatory diseases, or across even spatial transcriptomics imaging data. So sort of the episode that we held in the last session with one of my colleagues was relating to sort of pulmonary fibrosis disease and really how are we able to identify different macrophage populations. So you'll sort of see these different workflows on how you're able to actually perform the analysis, and it's quite useful to be able to see the case studies live and how you're able to walk through and use the BBrowserX system. So some of the, I guess, insights that we were able to gather from that specific case study was how we were able to identify 3 macrophage subpopulations. So macrophages that might express specific targets like SPP1 high expression. Some other ones might have FAP-P4 high expression, and these really determine on how patients actually respond to therapies or how their sort of resolution of inflammatory sort of cytokines in the lung. We were also able to verify how these different cell types and gene expression changes across different lung conditions. So some of the results from that case study will show sort of samples from healthy donor, samples relating to idiopathic pulmonary fibrosis, and then sort of comparing these differences across different lung regions as well. Some other information or other features that also were covered in that session were how to actually perform cell-to-cell communication analysis. So really understanding ligand binding interactions within sort of this microenvironment in the lung, and how this sort of translates to how there's resolution of inflammatory responses and how these different macrophages might interact with other cell types like fibroblast cells. So if you're really interested in these type of analysis workflow, or just how these different cells communicate and you want to be able to run that on your data, we do have within our YouTube channel all the recordings of these previous sessions as well. So the agenda points for this session, as I mentioned, it'll mainly focus on sort of stage 4 breast cancer and really looking at metastatic precursor sort of specific targets relating to these sort of cancer cells. So in the first part, I'll try to sort of cover an introduction of BBrowserX Pro, like as in terms of the workspace or the UI and how you're able to navigate these different features, as well as how can you connect the public data into BBrowserX Pro. Within that first session, we'll really try to identify malignant subclones and how to define these transcriptional states within these cancer cells. So really, what sort of feature will help us identify these mutational or these copy number variations in the cells that gives us the indication that this is sort of neoplastic cells that we can even subcluster further into different transcriptional states. The second part is from there, can we isolate this population and actually look at what are the pathway level features from these cells and how are they compared to sort of distant metastasis, so different sampling sites in the tissue. And then the third sort of final aim will really try to look at how these different sort of precursor cells have these different cellular transitions. So is there a way that we can track pseudotime subclonal differences based on sort of pseudotime analysis? So that's another feature that we have within the system that I'll sort of walk you through how to interpret the results and how to actually run the analysis in the system. So I guess before I go into the live demo, I do want to give some background if you're not working currently in this area and you haven't really worked on really defining what sort of metastatic precursor cells are and how can you actually define these transitional states. Even if there's a lot of sort of therapeutic advances within sort of oncology or different cancers, stage 4 breast cancer remains sort of a clinically intractable disease with really high rates of mortality. And this is really primarily due to the recurrent metastasis and therapeutic resistance that you see within these patients. These clinical failures are not merely sort of stochastic, but they are driven by sort of the evolutionary plasticity that you see within the tumor microenvironment. This might include what we would sort of term as intertumoral heterogeneity, which you often see within the sort of microenvironment, which is mainly defined by the coexistence of these genetically and transcriptionally diverse subclones within a single lesion. This heterogeneity might enable sort of paths of adaptation. So you'll normally see specific cells within this sort of lesion where they might have some hypoxic specific sort of roles, or they might have an inflammatory role, or they might sort of be part of sort of this extracellular matrix as well. If we're able to really characterize the intertumoral heterogeneity, which is something that we'll try to answer within these different functions, we can really understand how these drug-tolerant persister cells actually sort of are involved within this process. So really looking at rare or transient cell states of these cancer cells that might have some indication that they're actually tolerant or they don't respond well to certain treatments, and how do they actually adapt, and is there any pathways that gives us sort of some idea of their quiescent cells, if they're sort of stress resistant cells, or if they're just undergoing some metabolic rewiring. So those are things that we'll sort of try to understand with the pathway enrichment analysis as well as the trajectory analysis that we can perform. So other things that I'll also want to mention is some of the analysis that was already performed within the public data set and what we know so far relating to the mutational landscape in this microenvironment. So normally from other studies or emerging single cell or spatial transcriptomic studies, we have been able to link very specific programs relating to hypoxia-induced transcriptional states, as well as other phenotypes that are more aggressive. And those would normally be termed as sort of EMT, which sort of are brought by stemness or neoangiogenesis, where you see sort of high expression of VEGF-A. or the upregulation of a target called ADM. So these are sort of genes that we'll also try to visualize within our UMAP to see these transcriptional changes across different regions of these cancer cells from different lesions. Okay. So I guess our main research question within all of these different aims that we'll try to do is really how can we define molecular features and evolutionary trajectories from these metastatic precursor cells? And can we understand how these cells might be drug tolerant across different treatments or even different sampling sites from this stage 4 breast cancer study specifically? So the study that we'll be using is this sort of longitudinal serial single cell RNA-seq data set from Otsuji, which really tried, what he did was sort of look at a longitudinal serial experiment where he looked at one patient, so cells from one patient, and sampled them before and after therapy. So he had pre, post 1, post 2, and then the metastatic therapy sort of time point. And then he also looked at these sort of distant peritoneal metastasis sort of regions as well. So we'll be using sort of this data to really look at other questions and really diving deep into how can we do subclustering or look at subpopulations of these cancer cells, and can we identify these different functional states. So that will sort of be the focus. So in order to find this study, just to give you an idea, when you access the BioTrain ecosystem, you can go directly into the Talk to Data repository. So this is where we hold most of our curated data sets in the system. You can go into the study tab, and this is where you can either enter the sort of GSC ID of the study. So in this case, this would be this study. So I'm just showing you how you're able to input the GSC ID or the title of the study as well in the system. So when you actually find the study, what you can do is search through the whole Talk to Data system. You'll be able to explore that individual data set and get an idea of what metadata we currently have within the study. And let's say you're not only interested in this breast cancer data set, but you just want to see how many studies we have in the Talk to Data system relating to breast cancer, and also studies that might have like fibroblast cells or treatment data. What you can do is add that information in the ontology filtering to be able to identify very specific breast cancer data sets. You can then, after adding the condition, let's say I'm focusing mainly on breast cancer data sets, I can then include studies that have specific information on treatments or even treatment response. So I'm able to actually look at these different treatment response sort of differences across these lineages or these cell states. And that will give me sort of what studies we currently have in the system with this information. So you'll see that we have 13 studies relating to just breast cancer that have these different treatment response metadata that you can use. If you're not interested in treatment response and you just want to see how many studies we have within breast cancer, you can simply apply the search with just breast cancer, and you'll see that we have over 150 studies in this area. So that's how you're able to sort of explore public data sets and sort of mine the Talk to Data system. Now, when we actually explore the study, this data set will be directed into our main single cell analytics platform, which is termed BBrowserX Pro. You'll be able to see all of the metadata associated with that study, so including any QC parameters, any metadata relating to the annotations from the author, which might be a bit limited compared to what we can actually run with the meta-reference cell type prediction tool. And you can actually go through other information like the different treatment time points, for example. So this is where we'll start. So I guess the first, if I go back to our slides, the first question is really how can we identify cancer cells where it's not only mapped based on sort of gene expression or transcriptional gene analysis, but you're actually being able to determine if there's any aberrant or copy number variations within these different cells. So in order to be able to run that analysis, we do have the infer-CNV functionality within the BBrowserX system. So this feature can be found here under the cancer cell prediction model. And what is this approach actually doing? So this approach really tries to identify malignant cells from the actual study. It tries to group them based on the gene expression profile to form these pseudocells. These pseudocells really help to reduce any noise, any sort of signal for detecting any copy number variations within the cells that actually can distinguish from cancerous from normal cells. So in order to really do that, what you would have to do is actually either just run the model, the existing default model. So if we go into the cancer prediction, you can create a new analysis and you'll be able to select, okay, what main cancer tissue am I working with? So since this is breast cancer, I'll choose the carcinoma cancer type. And then you'll have two different options. One, which is the automated model, which is just going to sort of try to run all of the sort of pseudo counts across all these different cells. And or you can choose the manual selection, which is mainly based on sort of what cells would you term as normal, and it will use that as sort of a base to be able to then compare the different number company number variations that we would normally see, and if there's any sort of enrichment within these cancer cells based on these variations. So that's something that you can select which ones are your actual normal cells, so your I guess the non-cancer cells, and then which ones are cells that you're not really sure, but you want to really define them and see if they're actually neoplastic cells. So this is something that you'll need to sort of select. From here, you can add other advanced parameters if you need to add some subject information or a different embedding. You can also add that information in the output. Okay, so some sort of results that we can show you here in terms of what we see within the cancer cell prediction is that it is able to identify which cells are actually cancerous, and this is actually sort of it coincides with what the author actually found as well. So this is quite reassuring that you're able to actually validate all these results and you're able to run similar features that the authors did, but without any sort of, I guess, code or any notebooks that you need to actually run in the system. So from here, let's say we want to really understand these results further. So one thing to keep in mind when you run the cell type prediction, you'll have these different results. One, which is how you can stratify these malignant subclones. So this is based on these sort of copy number variations and how we see them across different chromosomes. Across these different lesion sites. So if I go back into the biotrain system, I'm able to go into the actual heat map view where I can see which chromosomes actually have very specific amplifications or deletions, and you're able to see that there is very clear differences based on the expression level of these subclones compared to the normal sort of cell types. So we can see that, for example, if we go deeper, we can see that there's specific amplifications or deletions within chromosome 1, chromosome 8, as well as chromosome 17 that you see these sort of, I guess, large indications. What can we tell from these different chromosomes? So normally these, just comparing from other studies, you're able to find usually oncogenic specific genes like MYC, sort of 𝐒𝐬𝐵B2 within sort of the 17 chromosome or the chromosome 17. So these are specific sort of analysis that you can gather is what can we see across these different chromosomes, and what have other studies identified as are there specific targets or oncogenic genes that are normally found within these chromosomes, and can we get more information on these specific cancer cells. So this is sort of one way that you can start gathering some information on these different malignant subclones. From here, there's another sort of visualization as well, which if I go into the slides, this is sort of a phylogenetic tree, which is showing you the subclonal architecture. And this is also mapped based on the CNV score that is sort of run from the actual prediction result. So some ways that you can sort of interpret these results when you're looking at the subclonal architecture is that you'll have these sort of nodes, which is sort of the actual subclone. So each node is a subclone. The phylogeny sort of structure represents really the evolutionary relationship among these sort of different cancer subpopulations and how there's different transcriptional divergence. So this is something that we'll need to really look at into further when we run the pseudotime analysis. Can we see these different lineages actually mapped on the UMAP? And can we identify these subpopulations of these cancer cells further? So this is just sort of the first step. If I go back now into our main UMAP, so let's say I want to go into my different clusters that I have within the different UMAPs. So these are our cancer cells that were determined from the actual cancer cell prediction. Let's say normally the cancer cell prediction, all that you will see is sort of the CNV score. So these are our cancer cells based on this sort of higher CNV, so these higher aberrations that we see within these cells. And if you want to then subcluster this population, let's say we want to sort of look at only these cancer cells, we can either use the lasso tool where you're lassoing this region further, and you can run further subclustering on these cells to identify if there's any subpopulations within these cells that have very specific transcriptional changes. So that's one way that you can do it. Another way is that you can also run cell type prediction to be able to train on these different clusters. So let's say I was able to subcluster the specific population, and then I'm able to sort of run further clustering analysis, which is going to be allowing me to run either Leiden or Louvain and look at specific subpopulation of these cells. This is where you would be able to do that. So if I go into some of my metadata that I've run previously, I can actually go into my Leiden clustering, which actually shows me these different distinct cancer cell subpopulations. Let's say I want to actually identify what are the signature markers that are really differentiating these different clusters within this cancer cell group. And are there any indications that these might be hypoxic, this might be an aggressive form with specific targets or other inflammatory processes. So if I want to look at what are the marker signature genes within each of these clusters, I can actually run this feature as well, which is really going to determine what are the gene expression level and the specificity within each of these clusters and what are their gene set markers. Another sort of option here, if you just want to be able to run cell type prediction and see if the system is able to identify some cancer cell population differences, this is where you can either run the personal reference algorithm. So this is where you're training your own you have a specific reference model data sets within the system that you've uploaded, and you have specific annotations from those studies internally. So you want to be able to transfer those cell labels onto this new data set that you're analyzing as well. So that's a personal reference model, which it has different methodologies or approaches that you can actually run. But you will need to actually select which ones from your sort of database are your actual reference data sets for this specific model. So you'll need to actually upload those data sets as well. If you want to use our public data, let's say we want to use all those 150 studies that are relating to breast cancer and especially specific sampling sites. So there might be other studies that have these metastatic sites or other information relating to these cancer cell metastatic subclones. So what we can do is actually customize the reference model from our database. So one way that you can do this is to select specific studies from our Talk to Data system that relate to the cell types that you're looking at. You can also relate to specific tissues, so sampling sites that you're mainly focusing on within the breast tissue or, for example, the metastatic site. And you can also look at specific conditions. So one thing that you can add here as some of the filters, let's say I want to focus on breast cancer. And I can actually include very specific types of breast cancer. Some of them might be sort of specific, like HER2 positive breast carcinoma, or some might be luminal A, so different subtypes that you might want to include. Or you can just include all these different breast cancer subtypes within the study. The other option here is where you can actually select the type of tissue region. So I want to make sure that this is selecting tissue within the breast. And I can also add other sort of sections as well that might be focusing on like the lymph node. So there are cases where if the study specifically looked at, let's say, the mesenteric lymph node, which is the case for this study, I can include data sets that might have information relating to that as well. So once I have those studies, it's going to show me how many studies were actually able to coincide with my specific filters that I've added from the Talk to Data system, and I can then predict. So that will run directly within the system. It will be able to show you what each cluster is defined. So let's say if I want to show you these results a bit further in the database, what are you able to actually see? So within every cluster, especially within our cancer cell subgroup that we were able to define from the INFER-CNV analysis, we're able to see that there are specific clusters that are definitely sort of identified as neoplastic cells. But what else can you actually gather from the meta reference is the core markers that are actually shared across these different public data sets. That might give you an indication into what type of cancer cell subpopulation this is. So for example, let's say I want to go deeper into this result. What can I gather from here? I can see that these specific studies were able to define these different neoplastic cells based on the core markers that are shared to actually label the cells and how these actually correlate with our signature markers that are ranked within this cluster 1. So cluster 1 is this sort of specific cancer cell subpopulation within this region. We are able to also confirm from the cell type prediction across other studies that this is an actual neoplastic or a cancer cell. And we can actually see all of our signature target genes for that specific cluster 1. So if you want to actually view these genes to get an idea how they're expressed or where they're expressed specifically, you're able to look at each individual target as well. You can also look at how these are ranked, so how the log2 fold change is weighted across these different genes in this cluster 1, or you can input other genes as well to really define what population or what type of cancer cell state this is in your data set. So this is one way we can go deeper into these subclusters, so we can gather all of these different marker genes across the different clusters in our data set to really understand. So if I want to see, let's say I want to see another different cell type population, I can go into those clusters in my data set and I can actually see what are the marker genes within that population as well. Okay, so that's in terms of how you can perform meta reference. Let's say I want to focus only on these clusters, and now that I know some of the genes that we have in the system, I can actually label these cells specifically. So let's say I want to look at this specific sort of cluster or cluster 1, whichever cluster you're actually labeling, and you want to identify what are the sort of marker genes. So this is where you can go back to the meta reference and get that list of marker genes and then relabel these clusters as well. So some of the examples here on how that would look and how you can annotate is, let's say we choose this cluster 8 group or this cluster 1. What I can do is create a new annotation for this cluster, and what I can do is also append it to an existing group. So if I want to add this level 2 I guess, labels to these cancer subgroups, I can add it to the original meta reference prediction list. And I can add what specific label am I adding to this cluster 1. So I can actually sort of say that this is sort of 82 positive, or whichever gene is sort of identifying this population. So once you have that, or you add that specific sort of list, what it'll show you when you add that list is that specific annotation that you've added as well. So you'll be able to create those labels specific to you. So one example on how I was able to go through these different clusters here that I had based on the prediction or the marker genes is to find really specific groups based on those marker genes. So what marker genes are relevant to like hypoxia adapted cells, for example, the TNBC, which is mainly aggressive specific cells that have a really high expression of CDA. Hypoxia adapted would normally have like an ADM expression as well. So these are how you can actually annotate and then sort of identify these different populations. So if I go into the other analysis, so how can I actually visualize other targets? And this goes really into our aim 2, which is really how can we define these different metastatic precursor sort of transcriptional changes, and can we look at specific genes that might give us indication on these differences, and how can we visualize this data? So in order to do that, once you have these specific subclustered cells, and let's say we only want to focus on these specific populations, I can select them from the list. I can subcluster them. And then I can try to sort of run other visualizations. So let's say if I'm bringing up one of my gene set lists that are specific to these metastatic precursor genes, I can specifically look at each individual gene and how it's expressed within these different cell populations or these different regions. So for example, this TACSTD2 gene is mainly expressed within this sort of basal-like EMT region. If we look at 𝐕M, we can also see exactly where it's expressed. So 𝐕M is mainly enriched within this calf region that has also the expression of PRSS35. And how can we visualize this data? So we can visualize it by looking at a summary across these different genes, which is going to be mapped across a UMAP or this specific cell populations. And you can also input other metadata. So if you want to see the actual cancer cell label, you can add your own label based on how you labeled the cells, and you're able to see that cluster as well. So you can actually define, okay, this is related to this subcluster, or you can also input other information like the actual samples. So if you want to look at these differences from these different sampling sites or these different pre and post treatment differences, you can also overlay that information. So you can see that there's specific genes like the LOX gene, that there's a high enrichment within this specific cluster that also has these metastatic cells as well. So you can actually go back and get the information or relevance on this UMAP if you go back into the original UMAP, and you can also select the sample ID to actually see what cells are relevant to these different regions. Okay, so that's one way you can visualize data. Another quite important way that you can do this is by looking at cell proportions. So if you want to look at how these different proportions or these different cell types that I was able to annotate from the cell type prediction and even go deeper into transcriptional changes, I can select my cancer cells, level 2 annotations, and I can look at how these proportions change across different samples or different pre and post treatment responses. So what we can see here is within my different cancer cell subpopulations, I'm able to see that mainly the metastatic sort of precursor cells are relating to the circulatory cells. So they have a really high expression of these specific genes. And you can also see that they're also relevant within other regions as well. So these aggressive specific genes, you can see them as well within the hypoxia adapted sort of group as well, where we see these metastatic sample cells mainly sort of relevant within this region. So that's how you can sort of gather some information on that, as well as different sampling sites. So if you want to see differences in the sampling sites as well, you can also overlay that information. Okay, so that's how you can visualize the data. Now, the next step is really how can we take this further and looking at different pathway or functional changes. So this is where we can move from the meta reference and actually visualizing different genes that are expressed across these different subpopulations. But we can actually select from our list specific groups that we want to identify how they're differentially expressed and are there any pathways that are showing these functional differences. So in that case, when we go into differential gene expression, you can create a new analysis here, and you can select what specific statistical method you want to use from your data. And how you're able to select those labels. You can use the composition chart here, which allows you to view different levels of the data, especially if you're running different cell type comparisons within a specific sort of treatment group. So this is where I can go into my composition and I can start adding the specific metadata that I'm interested in. So one is my cancer cell, and the other one is relating to the actual treatment. So my actual sample group ID. Okay. Within this, what I can do is actually select what cells I want to compare. So let's say I'm interested in these sort of hypoxia adapted tumor cells. I can compare how it's different within pre and sort of metastatic post-treatment analysis. And this is how I can gather how these hypoxic cells are actually very important in driving these transcriptional changes. So this is one way where you can select like the pre adapted hypoxia cells, and then my second group can be the metastatic cells here. So if there's very low number of cells, this is something that you'll need to keep in mind as well, that you might need to select another group to actually sort of increase the cell group, or what other population might be sort of equivalent to that as well. So if I go into the results here, just to show you what you're able to see. So I was really interested in really analyzing these hypoxic adapted cells and also how they differ from the circulatory cells, because these were the ones that actually had a lot of abundance of these metastatic sort of sampling site treatment sort of cells within these groups. So if we view the results, what we're able to see clearly is what are the top markers that are upregulated or downregulated. So you can actually change the direction on what you're actually focusing on, and you can select to see the top 20, for example. So these are very important genes that we see again, like the tax SCT2, which is normally seen in these hypoxic-driven cells. You can also see other targets as well, like KRT81, which is also another very important gene. And you're able to visualize this as well in a heat map view. So one way that you can do that is, for example, here I'm able to see these differences in the pre-treatment, so the pre-treated hypoxia adapted cells and how they actually change into the metastatic site. So we can see very specific genes that really have a role in the transitioning of these different cells across these different treatment or these different stages of the cells or tumor cells. Okay. From here, another thing that I can sort of quickly, because I know we're quite on time, is sort of what are the actual functional pathways are that are relevant from this differential gene expression analysis. So if we go into the differential expression, you'll see that from these specific results, you're able to run an additional GSA enrichment sort of test. And this could be from pathways that are related to biological processes, molecular processes, or even reactome processes. So we can really tie into how these genes are relevant, and are there any genes that also are showing up across these different sort of pathways within these samples. So one example here, when I ran the biological processes, what I'm able to clearly see from the first one is that there is sort of an enrichment of this sort of positive regulation of T cell mediated immunity. So how do these sort of immune cells interact with these tumor cells? We can also see other sort of pathways. So if I go back into the slide, we can see that there's also a cellular response to starvation and cellular respiration. So this is quite important because this really shows that these sort of specific ADM cells, so these hypoxic cells, adapt. And they adapt as well when they reach sort of a metastatic cell subclonal region. And there's a lot of these different sort of pathways that might give us an indication in how these are distributed and how these cells can upregulate immune or stress adaptation programs within the tumor. So again, this is sort of the type of analysis that you can run and how this represents this statistical significance across these different enrichment pathways. Okay, so I guess the last part that I'll briefly explain is really how can you actually map this and look at these trajectory transitions within these different cells. So not only the clusters themselves or the genes that are differentially expressed, but really looking at pseudotime analysis. So mapping these transitional cell states within the system. So in order to do that, you can go back into the data set and you can actually access the pseudotime analysis feature, which is based on Monocule 3. You can select what specific cells you're actually running. And normally it'll actually require you to select what is your sort of starting node, or where do you normally see these transitions based on the gene targets that we were able to gather. So if we go here into Centroid 85, so these cells right here were mainly based on the hypoxic cells as well. So I want to see if there's any lineage change and any genes that are actually driving these changes. So some examples here, if I'm able to overlay my cancer cell annotations, I'm able to see how these sort of change across different populations of these cancer cells. And I can also mirror the same targets that I was able to find also from the differential gene expression analysis. So I can see exactly how these different genes are enriched within specific cell sort of subpopulations or these subclonal differences, and which ones are actually driving these changes. So this is another way that you can also run, or you can also identify these transcriptional subclones within the metastatic site, and you can overlay these different sort of sample names or sample IDs to really look at the actual metastatic cells as well. Okay. So I guess from here, we've covered many different features, and some of the results that we were able to gather, just as a summary, is that we were able to see these hypoxic adapted tumor cells that had an expression of ADM positive gene, and these exhibited sort of a higher proportion within the metastatic site or the sort of treatment group or the sample group that have other sort of phenotypes. So these might have functions related to metabolic reprogramming or other invasive gene expression, or also differentiated profiles from circulatory metastatic clones as well. The second part was really looking at pathway enrichment. So this was an additional feature that you can run after performing differential gene expression, which gives you information on specific pathways that might be related to oxidative metabolism or immune evasion, like the T cell immunity pathway that was sort of enriched within that population. And the third sort of part was really how can we even look into this further when we're running pseudotype analysis? So can we define these sort of trajectory cell state differences, and can we then save that metadata? So if there is a case where you want to save the cell states, you are able to do that, and you should be able to see the result in your metadata category. Similar to how I'm able to see my states that I was able to run. And I can then use this to look at proportional changes across different groups as well. Okay, so now that we're done, my colleague will be sending the poll just to get an idea of this sort of case study was useful for you, if it allowed you to really understand sort of the workflow of BBrowserX Pro when you're trying to analyze these different tumor cells. How can you subcluster them? How can you use different features to identify these transcriptional changes? And really just to get some feedback from you if there's other features that we don't currently have, but would be really useful for your workflow as well. So really, if you have some time, this would be great to really gather some feedback from your end as well. Okay. So we'll give some minutes for users to add that information or to add sort of their feedback onto the system. And again, if there's any questions that you might have relating to your data, feel free to always reach out to our support email, and we're happy to walk you through these features where we can go through the data that you're working on specifically. Okay, so thank you very much for joining. I'm really happy to see users joining this call. And again, next week we'll be having another single cell exploration club that we can dive deeper into other case studies. Okay, so thank you so much for joining.